So let's talk about the first process of glycolysis. And glyco means sugar, lysis means break. So we are literally going to take our glucose molecule and break it in half. I have a quick overview of glycolysis. Glycolysis is going to occur in the cytoplasm of the cell. Mitochondria are not even needed. No, we don't need mitochondria yet. What we're going to do is we're going to take our glucose molecule, and in order to make money, you got to spend some money. So we're going to spend 2 ATP, which is going to make our glucose molecule unstable and break it in half. So we started out with one 6-carbon sugar called glucose, and we're going to end up with two 3-carbon sugars called pyruvate. And you are going to learn the name pyruvate. You don't have to memorize its whole chemical structure, but you do need to memorize that it has 3 carbons. We have to do some in order for us to take our glucose molecule to create the structure of pyruvate, we have to do some moving around of electrons. So during that process, we're going to produce 2 NADHs. So start a little column in your notes because we're gonna keep track of how many NADH we produce throughout this whole process. We're also going to produce 4 ATP. Now, if we remember, we gained 4 ATP, but we spent 2. So our net profit is 2 ATP molecules from glycolysis. So we're going to have a column of ATP for our notes as well. Let's just talk about what we've got. So we've got, at the end, we have our 2 pyruvate molecules, our 2 NADHs, and our 2 ATP that we've profited so far. All right, let's look at a more detailed diagram of what's going on here. So here we have our 6-carbon glucose molecule. We're going to take that 3rd phosphate group from one of our ATP molecules, and we're going to stick it on the end. We're going to take this ATP, stick that 3rd phosphate group on the end of our 6-carbon sugar. Those phosphate groups repelling each other makes this unstable. That's going to break into our 3-carbon sugar that has the phosphate group attached to it. We can move that phosphate group off. This is where we're going to produce our NADH. We're also going to produce our 4 ATP molecules as we move atoms around on these 3-carbon sugars to end up with our 2 3-carbon sugar pyruvates. Okay. This process is called substrative phosphorylation because we're using an enzyme to physically move one phosphate group from one molecule to another. All right, so importantly, glycolysis does not need oxygen. At the end of glycolysis, we have our pyruvate. At this point, we need to make a decision. So along this decision tree, the cell needs to decide, is there enough oxygen present to go on with, or is there not enough oxygen and we have to take a different biochemical pathway? So with oxygen is aerobic respiration, meaning with oxygen. Okay, and that's where we're going to go do, so we did glycolysis, we're going to do the oxidation of pyruvate, we're going to do the citric acid cycle, and then we're going to do the electron transport chain. But if there's not enough oxygen, we have to take this other pathway, then we're going to call that process fermentation, and we'll go through the details of this at the end. We're going to talk about the aerobic respiration pathway first. One of the problems that we have at the end of glycolysis is that we've taken our NADH, or sorry, our NAD plus, we've given it some electrons, 2 electrons and a hydrogen, and that's going to make our NADH. But if we use up all of our NAD plus to produce NADH, we have to have a mechanism to reform NAD plus so that we can continue glycolysis. So we don't have that yet. We are going to talk about when this happens in aerobic respiration, and we'll talk about when that happens for fermentation. That there has to be a mechanism to take our NADH and convert it back to NAD plus. Okay, so if you have your guided notes, now's a good time for us to pull out part 2 and look at that question number 1. This next clip will show you a table that you should be putting in your notes. Find a blank piece of paper that you are going to add this table to, and you're going to include more information as we go through the 4 stages of cellular respiration. So for the cellular respiration, you are going to memorize the chemical equation. We are going to start with 1 glucose molecule, C6H12O6. This means there's 6 carbons, 12 hydrogens, and 6 oxygen atoms in this glucose molecule. We also need 6 oxygen molecules. 1 glucose and 6 oxygens are our reagents. These are our starting point. At the end of the equation, we are going to end up with 6 carbon dioxide molecules, 6 water molecules, and then energy in the form of both heat and ATP. So we've talked about the process of glycolysis. So with our equation up here, let's just do some bookkeeping of what we've used and what we've produced. During glycolysis, we take our glucose molecule and we split it in half. So we've actually used up our 1 glucose molecule during glycolysis. So far we have used no oxygen. We have produced no carbon dioxide, and we have produced no water. But we have we've gained 2 ATP molecules, right? We used 2 to produce 4. That means that we've profited 2 ATP molecules. So we'll add that to our equation right now. All right, let's have another look at this glycolysis. So with my baby toys, what we're going to see here is we have our glucose molecule with 6 carbon atoms. And then I've got my ATP representatives here with my A base, my ribose sugar, and my 3 phosphate groups. I've got 2 of those. I've also got some ADP floating around in my cell and some inorganic phosphate groups. Okay, so what our ATP molecules are going to do first is they're going to transfer their 3rd phosphate group onto our glucose molecule. That makes the glucose molecule unstable because these phosphate groups are repelling, and that is going to help that sugar molecule then break in half. Okay, what we can then do is take these phosphate groups, move them back onto an ADP to remake some ATP. Do the same thing over here. Okay, now there are some electrons that are binding these carbons and some other atoms onto this that we're not showing. So during that rearranging of our 3 carbon sugars, so we end up with 2 pyruvate, we're also going to allow the formation of 2 more ATP molecules. So we're going to end up with a total of 4 ATP molecules. Remember, we spent 2. We are going to end up with our 2 pyruvate molecules, and then the thing that we're not showing is our NADH.